In this video, we'll talk about the kidney's role in maintaining the body's pH. The blood pH must be maintained at a narrow range of 7.35 to 7.45. The kidney is one of the two organs, the other being the lungs, that play a role in achieving this. The kidney accomplishes this task through the regulation of two ions, bicarbonate and hydrogen. To raise the pH, the kidney will reabsorb bicarbonate from the tubular lumen into the peritubular capillaries, which are an extension of the efferent arteriole, wrap around the entire tubular system, and eventually lead back into the renal vein and into the body circulation. The kidney can also excrete hydrogen from the peritubular capillaries into the tubular lumen, where it will travel the nephron and eventually be excreted into the urine and leave the body system. To do the opposite and lower the pH, the kidney will decrease the rate of both of these actions. The major parts of the nephron that will be active in these processes are the PCT, the DCT, and the collecting duct. The PCT will reabsorb 80% of bicarb filtered through the glomerulus, so we'll start there. Now that we're in the PCT, our two goals are to reabsorb bicarbonate and excrete hydrogen. The only exception to this would be in conditions of blood alkalosis, where the rate of these processes would be decreased. So let's get familiar with this equation, as this will be the starting point of all acid-base regulation in the kidney. Carbon dioxide and water are going to combine via carbonic anhydrase type 2 into the carbonic acid, which will spontaneously dissociate into a bicarb ion and a hydrogen ion. Now remember, our two goals are to secrete hydrogen into the tubular lumen to be excreted in the urine, as well as reabsorb bicarbonate into the interstitial fluid, which will eventually lead to the peritubular capillaries. Before we talk about hydrogen secretion, we need to mention this transporter, which is our sodium-potassium pump. This transporter is going to use ATP to kick out 3 sodium ions in exchange for 2 potassium ions. The energy used in our sodium-potassium pump is going to create a concentration gradient of sodium outside the cell that's going to cause it to want to come back into the cell. Now this is what's going to drive hydrogen secretion, as sodium now goes down its concentration gradient while hydrogen leaves the cell. In this way, hydrogen secretion is a secondary active transport, as the energy used from the ATP in our sodium-potassium pump is indirectly driving a hydrogen secretion. Now, as bicarbonate builds up in the cell, it's going to want to leave, and it's going to take a sodium with it, which is going to maintain the original gradient that was set up from our sodium-potassium pump. At this point, we accomplished our two goals of reabsorbing bicarb back into the blood and secreting hydrogen into the tubular lumen. Now the story doesn't end here for hydrogen, as it's going to combine with any excess bicarbonate that's still in the urine, and they're going to combine into our carbonic acid. Now this is essentially just a reversal of our original reaction, as our carbonic acid is going to be broken down by carbonic anhydrase type 4 into water and carbon dioxide. Now these products are going to be recycled back into the cell so that we can start up another round of this reaction. So as a result, there's no real net loss of hydrogen, as it's going to be continuously recycled in and out of the cell, but each time this cycle occurs, we're going to get 1 additional bicarb back into the blood. These processes account for 80% of bicarbonate reabsorption, and the other 20% will occur after we transverse the loop of Henle and make it into the distal convoluted tubule. Now that we've made it to the distal convoluted tubule and collecting duct, we have two different cells that will be more or less active depending on the blood pH conditions. Our type A, or alpha intercalated cell, is going to be more active in conditions of acidosis. Remember that type A is going to secrete acid, which will raise the pH of our blood while lowering the pH of our urine. Now, a type B, or beta intercalated cell, is going to do the exact opposite. It's going to be more active in conditions of blood alkalosis, and we can remember our type B cell because it secretes base in the form of bicarbonate. This will effectively lower our blood pH while raising the pH of urine. So in our type A cell, we know we want to secrete acid in the form of hydrogen ion, but how are we going to do that? Well, we can kick the hydrogen out through these two transporters, and they're both going to use energy in the form of ATP to accomplish this. The difference between them, the first being a hydrogen ATPase and the second one being a hydrogen potassium ATPase, is the second one will move potassium into the cell. Now remember, the potassium concentration inside of our cell is already high because we have this sodium potassium pump, which is going to kick out sodium in exchange for potassium. So the potassium isn't too comfortable inside of the cell, and for that reason, it has its very own transporter where it can leave on our basolateral side of our cell. In our type A cell, our main goal is to raise the pH of the blood while lowering the pH of the urine. So our second way of doing this after secreting hydrogen is to reabsorb bicarb. Now bicarb is able to go back into the blood where it'll raise the pH, and to maintain the negative charge, chloride's coming into the cell. Now, once inside the cell, the chloride isn't really too comfortable, because if you recall that the inside of the cell is negative at around negative 70 millivolts, you can see why the chloride, which is negatively charged, might want to leave, which is why it has its own transporter on the apical side. Now, a type B cell is doing the exact opposite of our type A cell. Instead of secreting our acid, we're going to secrete our base, which is bicarbonate. It's going to be exchanged for a chloride, just like it was in our type A cell, except this is occurring on the opposite side of the cell. It's happening on the apical surface. So our bicarb is going into the urine instead of being reabsorbed into our blood, and the bicarb will eventually be excreted. Now over here we have our acid, and instead of being secreted like it was in our type A cell, we're going to reabsorb it. And this is because in our type B cell, we're going to be active in times of alkalosis when our blood pH is high and we want to lower it. Our hydrogen transporters are also the same as they were in the type A cell, except they're on the opposite side. Now that they're on the basolateral side, we can reabsorb our hydrogen. So our first transporter in red is our hydrogen ATPase, and our green one is our hydrogen potassium ATPase. Now, just like in the type A cell, the potassium will be able to leave through its transporter. And the same is true for the chloride that came in from our bicarbonate chloride cotransporter, which is also called pendrin. So let's review what happened here. In our type A intercalated cell, we were in a condition of acidosis, meaning that our blood pH was too low. To combat this, we're going to reabsorb bicarbonate in exchange for chloride, and we're also going to secrete hydrogen through our hydrogen ATPase and our hydrogen potassium ATPase. In our type B cell, we're in a state of alkalosis, meaning our blood pH was too high. So to combat that, we reabsorbed hydrogen, which will lower our blood pH, and we're going to secrete bicarbonate. Now we're almost done, but we still have to take care of this free hydrogen that's in our urine because we don't want to let our urine get too acidic. So the solution for that is to combine hydrogen with ammonia to produce ammonium. And now this will be excreted in our urine. Now that we know how the kidney maintains the body's pH, let's test what we learned. The first question is, what enzyme catalyzes the reaction that produces carbonic acid from carbon dioxide and water? If you're thinking C or D, you have a 50% chance of being right here. But remember that one of these is going to work inside the cell to create carbonic acid, and one of these is working outside the cell to degrade carbonic acid into carbon dioxide and water. So the right answer for this question would be C, carbonic anhydrase type 2. This is going to be the form of the enzyme that's inside of our cell, and it's going to produce carbonic acid from water and carbon dioxide. Question 2, majority of bicarbonate is reabsorbed in which part of the nephron? Remember that one of these parts of the nephron is going to reabsorb 80% of all bicarbonate. Well, we can cross off a few of these choices, as the glomerulus is not even inside the nephron, so that's not doing any reabsorption. And the loop of Henle is also not active in bicarbonate reabsorption. Besides a small portion of the thick ascending limb, there's going to be no permeability to bicarbonate. So now between our PCT and our collecting duct, remember that majority of the bicarbonate is being reabsorbed in the PCT. Question 3, which of the following is an example of secondary active transport? Now remember that secondary active transport is when the energy produced from one reaction will drive another reaction. So if you recall from when we were talking about the PCT, how did we secrete out hydrogen? Well, first we used active primary transport via the sodium-potassium pump, which set up a concentration gradient for us. We were kicking out sodium, which created a force where sodium wanted to come back into the cell. So what we did is we coupled that force, allowing sodium to come back into the cell, with the secretion of hydrogen. And this was in our PCT. So the answer for this would be A. Remember that hydrogen secretion in the DCT was via a hydrogen ATPase or a hydrogen potassium ATPase. So that's primary active transport. And the sodium potassium pump that we were just talking about over here, that is another form of primary active transport, as it's directly using ATP. And choice D, potassium reabsorption in the collecting duct, what was the reason for that? We had potassium enter the cell where it was already in high concentration, and it left through a transporter on the opposite side of the cell, going down its concentration gradient. So that didn't use any form of energy. Final question. Bicarbonate is co-transported with which ion in the PCT? Well, we didn't really talk about calcium, so we can remove that choice. And we did talk about potassium, but that was with hydrogen, not bicarbonate. So we can get rid of that. Now, bicarbonate is co-transported with both sodium and chloride in the nephron, but which occurs in the PCT? That would be sodium. Remember that in our intercalated cells, that's when we're going to use chloride as a co-transporter, and that's why we have those special channels for them. But in our PCT, that's where bicarb is going to leave the cell, going into the blood with sodium and bicarb.